The transcription process in prokaryotes is quite different from that of the eukaryotes. In this video, I am going to explain about the transcription process in prokaryotes. Now, when we talk of the type of RNA which is found in the prokaryotes, there are majorly three types of RNA, that is mRNA, messenger RNA, tRNA, which is transfer RNA, and ribosomal RNA or rRNA. So we find all the three types, that is M, R and tRNA, present in the prokaryotes. Now, there is one enzyme which is responsible for the transcription process, and it is a complex enzyme which is having some subunits. So the enzyme which is responsible for the transcription process in the prokaryote is RNA polymerase, and it has several subunits which are functioning differently so as to achieve different results. So we have sigma over there, beta over there, rho over there, and beta dash over there, which are the subunits of the RNA polymerase enzyme, and they have a definite role in the process of transcription. Also, you have to remember that RNA polymerase enzyme will always bind at the promoter side, and also it will add the nucleotides in the direction 5' to 3'. So keeping this in mind, let us now understand that what or how the process of transcription is achieved in the case of prokaryotes. The first step is the initiation step. So when we talk of this step, this is the first step and it is known as initiation step. In the initiation step, that RNA polymerase enzyme, it binds to the promoter side. Over here you can see the promoter side of the DNA strand. So it binds to the promoter end of the structural gene with the help of sigma factor. So we have sigma factor with the help of which the RNA polymerase enzyme, this is RNA polymerase over here, and inside is the sigma factor. So this is RNA polymerase, and this binds to the promoter end with the help of this. Now the DNA as a result starts unzipping, and only the strand with 3' to 5' polarity, that is 3' to 5' which is marked in blue, acts as a template strand over here, and it will be coding for the synthesis of RNA. The second important step over here is the elongation. So after the attachment of the RNA polymerase with the help of sigma factor to the promoter, the unzipping of the DNA molecule starts, and further the elongation of the chain has to initiate. So what happens is the RNA polymerase enzyme then polymerizes the ribonucleotides in the 5' to 3' direction, because over here the strand which is going is the template strand is 3' to 5'. So the complementary strand will be opposite, that is 5' to 3'. And over here the last stage is the termination. So the third stage which we are talking over here for the transcription process in the case of prokaryotes is the termination process. Now in the termination process, what happens is the RNA polymerase reaches to the terminator sequence. The terminator sequence is downstream of the coding strand. So over here the terminator, now the RNA polymerase attaches itself with the help of over here to the terminator with the help of a rho factor, and It is also known as the termination factor, with which the RNA polymerase binds for transiently, that means for a short period of time, and this stops the process of transcription and the nascent RNA is released. So what is happening over here, if we in a nutshell, if we see, the process starts with the initiation, that is when the RNA polymerase enzyme binds to the promoter site with the help of the sigma factor. When it attaches itself to the promoter site, the unzipping of the DNA molecule starts, and it is followed by the process of elongation, where consecutively the nucleotides are being added, and it is in the direction of 5' to 3'. As a result, the RNA is being produced because the template strands actually codes for the RNA synthesis, which type of RNA will be produced. And on reaching when the RNA polymerase reaches the terminator end, the polymerase enzyme then attaches or binds itself to the rho factor transiently and attaches to the DNA, which eventually causes the stopping of the transcription process in the case of prokaryotes.